Okay, so now we're going to get into these extensions of Mendelian inheritance. So remember, Mendelian inheritance is where you've got a Punnett square and you've got a gene, and there's two alleles for that gene, and one of the alleles is dominant and one of them is recessive. So if you've got like the big A, you're going to have that phenotype. If you got the little a, you got that phenotype. And that works for lots and lots of genes, but that does not work for all genes. Think about just a lot of traits inside of humans, inside of your own body. Think about like your height. There's not just two heights for humans, like you're either 6 foot or 4 foot. It's this big diversity, right? Think about skin tone. There's not just like, you got like dark skin or light skin. There's all kinds of things in the middle. And so there are certain traits inside of every organism where there really are just two options, and that would be Mendelian inheritance. But there are many other traits where it's going to be something that is an extension on that. And so again, Mendelian inheritance is summarized with these two laws, which was law of segregation, that every gamete has got only one allele for each gene, and then this law of independent assortment, that whatever you inherit for one gene and allele combination has no impact on a separate gene, that all of them act alone. But there's going to be situations where it's not that way. And so there's lots of them. There's a bunch of different scenarios that can happen. And so these tables, there's 2 slides here of these tables. These do a great job of just summarizing what those are. And we're going to go through them one by one right now and just talk about them. But for each one of these, you should be able to describe in your own words, like 2 sentences maybe, what this pattern of inheritance would be. And so the flashcards you would maybe want to make are for these words over here in the very first column. And then the thing you'd want to put on the other side of the flashcard, the thing you would be able to say is this description over here on the right for each one. Okay, so we're just going to go down the line one by one, talk about what they are. So this very first one is just going to be Mendelian inheritance, where you got dominant and recessive alleles. And so the first question is just like, why does that even happen? Why is one allele dominant and why is one allele recessive? Like, what does that mean? And so in general, we call the dominant allele the wild type allele. This is the one that is going to be the most common. Because again, if you're going into a population where you don't know anything about those organisms, you don't know which one is dominant or recessive. And so the word that we usually use to describe just whatever is normal is wild type. Okay. And a lot of times these traits aren't going to be necessarily something really distinctive like flower color. It could also be like, do they have leaves? What shape are the leaves? Do the leaves have chlorophyll? Really like base things like that, right? But within a large population, there's always going to be diversity, and there's got to be some genetic basis for that diversity. And so that diversity we describe as genetic polymorphism. Poly meaning many, and then morph meaning different versions. And so there are likely going to be multiple ways that a trait could represent. And it depends on, is there like one gene controlling that trait or multiple genes controlling that trait? In this example over here was the color of these orchids. Those polymorphisms we tend to call mutants, mutant alleles. And most mutants aren't going to be successful. Mutants oftentimes show up in the population just from mistakes during DNA duplication. They could also be from recombination, meaning that you get like a new combination of alleles that didn't used to exist. In that case, they're probably not going to be harmful because it's the same alleles that were already there. They're just maybe swapped around in a new way. But in general, if they're recessive, usually that's going to do something bad to the functional protein. In some way, it's going to either be expressed less, or the version that is expressed is a less good version of that protein. And so oftentimes, whatever is not dominant is something that is less adaptive. And so if we look at some examples of recessive diseases, recessive human diseases, these are ones where there's an allele, it's recessive, and if you have that recessive allele, homozygous recessive, this is the sort of disease that you'd end up with. Cystic fibrosis is one you've probably heard of before. There's other ones here like the Tay-Sachs disease. And so all of these, it means that you'd have the recessive allele. And so why is that? Why is it that the mutant tends to be recessive? And so there's two reasons for that. And so remember that all organisms, you got a homologous pair, so you're going to have two alleles for every gene. And so you're oftentimes not going to get two recessive alleles. That'd be possible, but just really unlikely. Instead, you're probably going to get one recessive allele. And so if it's just one recessive, that means you also have one that is dominant or wild type. And so for many organisms, that one individual wild type allele is enough. That wild type allele is going to build a protein, and that protein will be present in sufficient quantities to just do its job. And so this one type copy is enough. The other option is that you have only one copy, but the organism's cells will recognize there's only one copy of that allele. And so it'll upregulate it. It'll make more of that wild type protein. And so in the end, you end up with the same phenotype. So in this example, if you got big P, you're going to get purple flowers. For this genotype, big P, little p, the heterozygous condition, there's only one dominant allele, but the organism still is able to produce a sufficient amount of this purple pigment toward the flowers are purple, even though you've got the recessive allele mixed in there within the chromosomes. And so when we've got these dominant alleles, they can become mutated, and they're either going to result in gain of function. So like they're a good thing. So because we have this new allele, there's some new phenotype that didn't used to be there. They could be dominant negative, which means that the normal protein is antagonized or downregulated or inhibited by the mutant protein. So somehow this mutant protein goes out and like attacks the dominant version. Or it could be that this mutant is a loss of function. And so now the mutant is loss of function, and then the remaining wild type copy isn't enough to compensate. So you end up just like not having that dominant trait. Instead, you're going to have some other version, which we're going to call recessive, but really it's probably like loss of that protein's ability to do anything. And so when that can happen, we get to a couple of different versions of what we can observe. And again, these are non-Mendelian patterns. So one of them is incomplete penetrance, and one of them is variable expressivity. As we go through this list, you'll see that oftentimes there's going to be like two extensions, two versions, and they're kind of sort of related. Okay, so incomplete penetrance and expressivity are kind of sort of related. In these examples, that means that you've got an allele, and that allele is different from the wild type, but it doesn't always penetrate within the population. And so that means that sometimes you're going to have individuals that have this recessive allele, but they end up with the wild type phenotype anyways. And so incomplete penetrance is when an allele is incompletely penetrant. It's incompletely dominant. It's incompletely expressed in the phenotype of all of the individuals within that population. And we measure this as like what portion of them are going to end up expressing that non-wild type phenotype. And then we would say it is either like 60%. This would mean that like 60% of the organisms that have that mutant allele actually have this mutant phenotype. The other 40% might, if you look at the chromosomes, have this mutant allele. They should have this other phenotype, but instead they present the wild type phenotype anyways. And so polydactyly is an example of this. Autosomal dominant allele. So if you have the allele for polydactyly, it's actually dominant. It's the dominant allele, but you don't always have it expressed within the population. So that means there's going to be some people who actually have this dominant allele for polydactyly, meaning they should have multiple fingers, not just 5, have like 6 fingers, 7 fingers. Or for whatever reason, they end up having 5 anyways. And so sometimes individuals carry the dominant allele, but do not actually exhibit the trait. And so again, incomplete penetrance is measured across the entire population. And it's where you've got the allele for a phenotype, but that phenotype isn't always expressed. Variable expressivity is describing the degree to which a trait is expressed. So here, with this one, we're saying that you've got the allele for this mutant phenotype. When you have it, you always express it at some level, but it might not be expressed to the same degree. And so in this example, we would say that, okay, well, they've got polydactyly, but this 6th finger is pretty small compared to this guy who's got 6 fingers, but the 6th finger is much bigger. And so again, penetrance across the entire population. Expressivity, we'd be talking about an individual. How expressive is that allele within this individual person? The environment can also affect gene expression. That's a really big deal. We're going to see that a lot. But changes in the environment can cause changes in a phenotype. And that could mean that you have two organisms that, from a genetic perspective, have exactly the same information, but phenotypically they're going to present differently because the environment is causing the phenotype to change. So an example would be coat color. And so Arctic foxes, if they are in snow, then they're going to have white coats. But then if they're not in as cold conditions, they're going to have brown coats. Another one is PKU. PKU means that you cannot metabolize phenylalanine, which is an amino acid. It's a big deal. So we screen kids for PKU now in the hospital when they're born. And if it turns out that they have PKU, then you can make changes in their environment and their diet that will keep them healthy. Even though if that was not being screened, they'd be sick. And so again, that's a change in the environments that affecting the phenotype. Whenever we're talking about how the environment causes a change, though, there is going to be what is called a norm of reaction. And so that would just describe how much the environment can change the phenotype. And so in this example, your options for coat color are white and brown, and the difference is the environment, but it's not like there's going to be foxes running around with like purple fur or something like that. There is like constraints. And so that norm of reaction describes the options that might be in terms of how that phenotype might change. So those two go together. So those are our first kind of like pairing, expressivity and penetrance. Okay, the next one is going to be incomplete dominance, overdominance, and codominance. These kind of all go together. Okay, so incomplete dominance would be first. Incomplete dominance is when the heterozygous individuals have an intermediate phenotype. Incomplete dominance, intermediate. Incomplete dominance, intermediate. So in this example, you've got individuals that are homozygous dominant, they're red. You've got individuals that are homozygous recessive, they're white. But then if you get heterozygous individuals, those alleles don't both, it's not that the red one is now dominant and the white one is silenced. Instead, they're both going to be expressed, and you end up with the phenotype that is intermediate, which in this case would be pink. If instead it was like black and white, maybe the heterozygous ones would be gray. So incomplete dominance is when there is not necessarily a dominant allele and a recessive allele. They're both being expressed, and because they're both being expressed, your phenotype that you observe is intermediate between the other two. That's incomplete dominance. Overdominance is when the heterozygous individual has greater reproductive success than either homozygote. And so it's also sometimes called heterozygous advantage. And so this means that for whatever reason, whatever this gene allele, the combo that we're talking about is, that being a heterozygous individual is more adaptive than either of the other two options for homozygosity. So that means that those heterozygous individuals, since they're more adaptive, are probably going to become the most abundant in the population. But if you think about that Punnett square in your head, a lot of the individuals are heterozygous, right? That means you're going to have like big A little a crossed with big A little a. You're going to get majority big A little a. Half of them are going to be big A little a, but you're also going to get another half that are going to be either homozygous dominant or homozygous recessive. And so in this example, if there is a heterozygous advantage, heterozygous individuals are going to be the most abundant, but homozygous individuals are also routinely going to be produced, because that's just the way that Punnett squares work. Because of the law of segregation, you're always going to end up with one allele inside of one gamete. And so an example with this is sickle cell anemia. If you have sickle cell anemia, it's going to shorten your lifespan. Your red blood cells do not work as well. They cannot carry oxygen as well, and it results from being homozygous recessive. But if you are heterozygous for the condition, you're actually going to end up being more resistant to malaria. And so in environments where malaria is common, which is a lethal sort of a condition, being sickle cell anemic, but just heterozygous, so you kind of sort of have sickle cell, but not all the way to where it's going to shorten your lifespan, is actually going to make you more fit for that environment than someone who is not malaria resistant, which would be big A in this case. And so overdominance means that there is a heterozygous advantage. Now we get to the next one, which is multiple alleles and codominance. These two go together. Okay, multiple alleles and codominance. Okay, so multiple alleles is exactly like what it says. That means you got a gene, and for that gene, there's more than just two alleles. So in normal Mendelian inheritance, you got like big A, little a, right? You got big C, big M, little m, whatever. There's two alleles. Well, multiple alleles means that you're going to have like big A, little a, and then like double little a or something. You're going to, usually we don't use the same letter. We would say different letters. But a good example of this is blood type. So with blood type, you can have the A antigen. That's the allele is A. You could also have B type blood, which means you got the B allele, or you could have both, where you got AB blood, right? So far, we still only got two alleles. It's just if you have two alleles, A, B, you're going to behave differently than if you were just A or B alone. But then there's an additional option, which is that you could also just have neither antigen. So you do not have the A or the B, in which case we say you have O-type blood. And so in this example, we've got this multiple allele thing happening and then also codominance at the same time. And so we've got multiple alleles in that you could have A, B, or none at all. That still counts for multiple alleles. Then we also have this thing called codominance. Codominance means that both of the alleles can present, they can both be active and represent their own phenotype. And that would be AB blood. And you might be like, well, how's that any different from incomplete dominance over here? Because with incomplete dominance, it was the same thing, right? Like you had the red and the white, and then you got both of them together. So now your heterozygous individuals are expressing both, and then this intermediate phenotype. The difference is that with incomplete dominance, the heterozygous individuals are intermediate between the other two. So it's like red, white, pink, black, white, gray. The third phenotype is in between the other two. With codominance, the third phenotype, in this case type AB blood, is like totally different from the other two. So type AB blood individuals act differently from type A blood individuals or type B blood individuals. AB is not necessarily intermediate between A and B. It's its own distinct phenotype. So like with those flowers before, if it's incomplete dominance, it's red, white, and then pink. If it was codominance, it'd be like red, white, and purple or something, like some completely different color that's not even like the red or the white. Okay, red, white, and blue, something like that. And so codominance is where the heterozygous individuals have a third phenotype, but that third phenotype is not intermediate between the other two. It is its own distinct phenotype that is not like the other two. And so that would be codominance. Next, we got sex influence and sex limited inheritance. These are also sometimes called sex linked genes. That's another option that we'll get to. So here it means that the inheritance pattern of a trait is going to have an interaction with the sex of the individual. And so remember that with normal Mendelian inheritance, we've got the law of interdependent assortment, which says that whatever happens with one gene has no bearing on the inheritance pattern of another gene, that they all act independently in any combination as possible. With both sex influence and sex limited, that's saying that no, like for boys, the inheritance pattern is going to be one way, but for girls, the inheritance pattern is going to be another way. And so the sex of the organism influences the inheritance pattern. And so first would be sex influenced. With sex influenced, it could be that it's dominant, the one allele is dominant in one sex, but it's recessive in the other sex. An example would be these things called skers in cattle. They're these little bitty growths that are on the skull. And so they're dominant in males, they're recessive in females. And so if you end up with SCP and SCA, skers could be present and absent, depending on if that cow is a boy or a girl. And so if it's a boy, then that means SCP would be dominant. But then if it's a girl, SCA is going to be dominant. And so that'll determine the inheritance pattern is male or female. That's sex influenced. Sex limited means that those are traits that occur in only one of the two sexes. So it's only going to work for boys or it's only going to work for girls. And This one's maybe a little bit more straightforward, which is be like ovaries. If you have an inheritance pattern for ovaries, you're going to see that inheritance pattern in females. You won't see that in males. If you have some kind of trait related to testes, you're going to see that in males. You wouldn't see that in females. Plumage in birds would be the same way. Birds, oftentimes the males and the females can look really different from one another. And so think about like cardinals, for example. The boy is red, the female is like a dull brown color. Think about chickens. The male roosters look very different from a chicken, egg-laying chicken, right? And so sex limited means that an allele is only going to show up in one gender or the other one. Again, that's different from sex influence, because of sex influence, it does show up in both sexes, but it's just that which one is dominant and which one is recessive is going to change depending on the sex of the animal. This is what those things look like. These are the skers, okay? And then inside of chickens, for example, the plumage that we would see inside of those birds. Lethal alleles. So we can have alleles that are going to be lethal if they are inherited. And so this kind of is going to sort out based on what kind of a gene are we talking about. And so most of the genes that are inside of all chromosomes, inside of the genome, are essential. And they do like really boring stuff. And so when we're talking about, oh, your genome and what do you inherit from mom or dad, you're probably thinking about like skin color and hair texture and height and I don't know, the way you act, behaviors, I don't know, stuff like that, right? Well, those are a very small subset of the genes that you have inside of your body. Most genes are going to code for stuff like, how do your mitochondria work? How do you build cell membranes? Do you have functional motor proteins and a functional cytoskeleton? Like really basic stuff that's going to be inside of the cell. And so about a third of all of our genes are completely essential, and they do just normal housekeeping day-to-day stuff. And if you mess those up, it doesn't really matter what the allele is, it's gonna be lethal. Those are going to be proteins that are at the core of multiple functions inside of your body. They do lots and lots of different things. And if you mess any part of them up and break that protein, it's going to have so many consequences that it's going to end up being lethal. And so for essential genes, mutations in those genes could very often lead to a lethal phenotype. And if it's lethal, that means that the embryo is not going to develop. And so people who have mutations that are lethal in these essential genes, we don't see them. You're not going to meet people that have those mutations because they wouldn't survive. They would never be born. And so that's usually the outcome, is the embryo is not going to develop. We could end up with lethal alleles or just changes in non-essential genes. So you could have non-essential genes that get this lethal allele, and you're like, well, how does that work? The gene isn't even essential. And it's because there could be a mutation that causes that gene to all of a sudden be problematic in a way that it wasn't before. Usually these are recessive, and it means that this gene that used to be inert is now lethal to you in some way. It's doing something really bad inside your body. We also have conditional lethal and semi-lethal. So conditional lethal, this can be things where it can kill the organism, but only under certain environmental conditions. So developing Drosophila could be killed at 30, but they'll survive if grown at 22. And so that means that almost all of their proteins are temperature-sensitive lethal alleles. And that isn't that surprising, right? If you heat up a little baby when it's trying to develop, it's probably going to mess it up. And so there are many temperature-sensitive lethal alleles. There's also semi-lethal alleles. So these are ones where it will kill some, but not all. And a lot of times it's going to be with the environment. But the reason that's important is because let's imagine that you were doing a cross, right? And you're collecting data in the lab, and you calculate your expected offspring frequencies for phenotypes or genotypes, and then you go count those offspring and your numbers don't match. It could be that the reason why you're not seeing certain phenotypes is maybe those phenotypes are semi-lethal and not all of those offspring are surviving, which may inadvertently initially make it look like you don't know what you're doing. You expected to see a certain ratio, you're not seeing that. It could be that those offspring were produced, but they just didn't survive until birth, and that's the reason that your numbers are messed up. And so, again, Manx cat would be an example like that. Dominant mutation affects the spine. shortens the tail in heterozygous individuals, but it's lethal if you got homozygous ones. And so a lot of times with these lethal alleles, it is easier in terms of our genetics calculations when the lethal ones are completely lethal, because then we'll know why that organism isn't present. So if you're ever looking at our population, you're like, why don't I ever see this particular phenotype or this particular genotype? That's probably why, is that it must be a lethal outcome.